Everyone, welcome to NCERT audiobook. Today we start the new lesson of the class 11th, chapter 9, Biomolecules. There is a wide diversity in living organisms in our biosphere. Now a question that arises in our mind here: are all living organisms made up of same chemicals, that is elements and compounds? You have learned in chemistry how elements analyze in performed. If we perform such analysis on a plant tissue, animal tissue or a microbial paste, we obtain the list of the elements like carbon, hydrogen, oxygen and several other, and their respective content per unit masses of the living tissue. If the same analysis is performed on a piece of the earth crust as an example of non-living matter, we obtain a similar list. What are the difference between the two lists? In absolute term, no such difference could be made out. All the elements present in the sample of the earth crust are also present in the sample of living tissue. However, a closer examination reveals that the relative abundance of the carbon and hydrogen with respect to the other element is higher in many living organisms than in the earth crust. How to analyze the chemical composition? We can continue asking in the same way what type of the organic compounds are found in living organism. How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue or a vegetable or a piece of the liver and grind it in trichloroacetic acid. Using a mortar and the pestle, we obtain a thick slurry. If we were to stain it through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically the acid soluble pool, and the second is retention rate, or the acid insoluble fraction. Scientists have found the thousands of the organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffix it to say here that one extract of the compound, then subject the extract of the various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, gives an idea of a molecular formula and the probably structures of the compound. All the carbon compounds that we get from the living tissue can be called biomolecules. However, the living organisms have also get the information, inorganic elements and the compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weight of a small amount of the living tissue and dry it. All the water evaporates and remaining material gives dry weight. Now if the tissue is fully burned, all the carbon compounds are oxidized to gaseous form, CO2 and water vapor, and are removed. What is the remaining is called ash, and this ash contains the inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, the element analysis gives elemental compositions of the living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analyzing for the compounds, give an idea of the kind of the organic and inorganic constituents present in the living tissue. From a chemistry point of view, one can identify the functional groups like aldehyde, ketones, aromatic compounds, etc. But from the biological point of the view, we shall classify them into amino acid, nucleotides, bases, fatty acid, etc. Amino acids are organic compounds constituting a amino group and the acidic group of the substituents on the same carbon, that is the alpha carbon. Hence, they are called alpha amino acids. They are substitutes methanes. There are 4 substituents group occupying the 4 valency positions. These are hydrogen, carboxyl group, amino group and the variable group, designed as a R group. Based on the nature of the R group, there are many amino acids. However, those which occur in the proteins are only for the 20 type of the R group. In this proteinous amino acid could be a hydrogen, a methyl group, alanine, hydroxymethyl serine. 3 of the 20 are as shown in figure. The chemical and physical properties of the amino acid are essentially of the amino, carboxylic and R functional groups based on the number of the amino and carboxylic group. There are acidic, glutamic acid, basic, lysine and neutral valine amino acid. Similarly, there are the aromatic amino acids. A particular property of the amino acid is ionized nature in the NH2 and COOH group. Hence, in solution of the different pH, the structure of the amino acid changes. Lipids are generally water insoluble. They could be simple fatty acid. A fatty acid has a carboxylic group attached to an R group, and the R group could be methyl and ethyl or higher number of the CH2 groups. For example, palmitic acid has the 16 carbon including carboxylic carbon. Arachidonic acid has a 20 carbon compounds including the carboxylic carbon. Fatty acid could be saturated or unsaturated. Another simple lipid is the glycerol with the trihydroxypropane. Many lipids have both glycerol and fatty acid. Here a fatty acid are found esterified with the glycerol. They can be then monoglycerides, diglycerides and triglycerides. There are also called the fats and oils based on the melting point. Oils have a lower melting point. Example, gingerly oil, and hence remains as oil in winters. Can you identify a fat from the market? Some lipids have a phosphorus and phosphorylated organic compounds in them. These are the phospholipids. They are found in the cell membrane. Lectinin is the one of the example. Some tissues, especially the neutral tissue, have lipids with the more complex structure. Living organisms have a number of the carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases, adenine, guanine, cytosine and uracil and thymine. When found attached to the sugar, they are called nucleosides. If the phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine and cytidine and nucleosides. Anidylic acid, thymidilic acid, guanylic acid and uridylic acid and cyclidylic acids are nucleotides. Nucleic acid like DNA and RNA consist of the nucleotides only. DNA and RNA function as a genetic material. Primary and secondary metabolites. The most exciting aspect of the chemistry deals with the isolating thousands of the compounds, small and big, from the living organism, determining their structure and, if possible, synthesizing them. If one were to make a list of the biomolecules, such as a list would have to thousands of the organic compounds, including amino acids, sugars, etc., for reasons that are given in section 9.10. We can call these biomolecules as metabolites. In animal tissue, one notice that is the presence of all such categories of the compounds shown in figure 9.1. They are called the primary metabolites. However, when one analysis point, fungal and microbial cells, one would be seen the thousands of the compounds other than these are called the primary metabolites, like alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scent, gums, spices. These are called the secondary metabolites. While the primary metabolites have identical functions and play known role in normal physiological process, we do not at the moment understand the role of the functions of all the secondary metabolites in most host organisms. However, many of them are useful to the human welfare. Rubber, drugs, spices, scent and pigments, and some secondary metabolites have ecological importance. In the latter chapter, you will learn more about these biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have a molecular weight ranging from the 18 to the around 800 daltons approximately. The acid soluble fractions has only 4 types of the organic compounds: proteins, nucleic acid, polysaccharides and lipids. These classes of the compounds, with the exceptions of the lipids, have a molecular weight in the range of the 10,000 dalton and above. For this very reason of the biomolecules, that is, chemical compounds found on the living organisms, are of 2 types. One of those which have a molecular weight less than the 1000 dalton and are usually referred to as a macromolecules or simply biomolecules, while those which are found in the acid insoluble fractions are called the macromolecule or biomacromolecules. The molecules in the insoluble fractions, with the exceptions of the lipids, are polymeric substances. Then why do lipids, those molecular weights do not exist 800 dalton, come under the acid insoluble fraction, that is macromolecule fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into the structure like the cell membrane and other membrane. When we grind a tissue, we are distributing the cell structure. The cell membrane and the other membranes are broken into pieces and from vesicles which are not water soluble. Therefore, these membrane fragments in the form of the vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool are represent roughly the cytoplasmic compositions. The macromolecules from the cytoplasm and organelles become the acid insoluble fractions. Together they represent the entire chemical compositions and living tissues of the organism. In summary, if we represent the chemical compositions of the living tissue from the abundance point of the view and arrange them the class wise, we observe that water is most abundant chemical in the living organism. Proteins. Proteins are polypeptides. They are linear chain of the amino acid linked by the peptide bonds as shown in figure. Each protein is a polymer of amino acid, as there are 20 types of the amino acid: alanine, cytosine, proline, tryptophan, lysine, etc. The protein is the heteropolymer and not a homopolymer. A homopolymer has only one type of the monomer repeating the n number of the times. This information about the amino acid content is the important as the later in your nutrition lesson. You will learn that certain amino acids are the essential for our health and they have to be supplied through our diet. Hence, dietary products are the sources of the essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make. While we get the essential amino acid through our diet, food, proteins carry out many functions in the living organisms. Some transport nutrients across the cell membrane, some fight infectious organisms, some are hormones and some are enzymes, etc. Collagen is one of the most abundant protein in the animal world, and ribosomal biphosphate carboxylase oxygenase rubisco is the most abundant protein in the world of the biosphere. Polysaccharides. The acid insoluble pellet also as a polysaccharide carbohydrate as another class of the macromolecules. Polysaccharides are the long chains of the sugars. They are thread, literally a cotton thread, containing the different monosaccharides as a building block. For example, cellulose is the polymeric polysaccharide consisting of only one type of the monosaccharides, that is glucose. Cellulose is the homopolymer. Starch is the variant of these but present as the storage house of the energy in plant tissue. Animals have another variant called glycogen. Insulin is the polymer of fructose in a polysaccharide chain, say glycogen. The right end called the reducing polysaccharide chain, say glycogen. The right end called the reducing end of the left end called the non-reducing end. It has the branch as shown in the form of the cartoon. Starch forms the helical secondary structure. In fact, of the starch can hold I2 molecules in the helical protein. The starch I2 is the blue in color. Cellulose does not contain complex helicals and hence cannot hold the I2. Plant cell walls are made up of the cellulose, paper made from the plant cell, and cotton fibers is cellulosic. Therefore, there are more complexes polysaccharides in nature. They have as a building blocks amino sugars and chemically modified sugars, glucosamina and acetyl glycosamina, etc. Exoskeleton of the arthropods, from example, have a complex polysaccharides called chitin. These complex polysaccharides are mostly homopolymers. Nucleic acid. The other type of the macromolecules that one would find in the acid insoluble fraction of the any living tissue is the nucleic acid. There are polynucleotides together with the polysaccharides and polypeptides. These comprise the true macromolecular fraction of any living tissue or cell. For the nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct compounds. One is the heterocyclic compound, and the second is the monosaccharides, and the third is the phosphoric acid or phosphate. As you notice in figure, the heterocyclic compound in the nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are the substitution purines, while the rest of the substituting pyrimidines. The skeletal heterocyclic ring is called as a purine and pyrimidine respectively. The sugar found in the polynucleotide is either ribose, monosaccharidic pentose, or deoxyribose. A nucleic acid containing deoxyribose is called the deoxyribonucleic acid, while it contains the ribose is called the ribonucleic acid. Structure of proteins. Proteins, as mentioned earlier, as a heteropolymer containing strings of the amino acid. Structure of the molecules means the different things in the different context. In inorganic chemistry, the structurally invariable refers to as the molecular formulae. Organic chemistry always write a two-dimensional views of the molecules while representing the structure of the molecules. Benzene physicists conjure up the three-dimensional views of the molecular structure, while the biologists describe the protein structure at 4 levels. The sequences of the amino acid, that is the positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structure of the protein. A protein is imagined as a line, and left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called the N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of helix. Of course, only some portions of the protein's threads are arranged in the form of the helix. In proteins, only right-handed helix are observed. Other regions of the protein's threads are folded into other forms in what is called of the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow rounded ball, giving rise to the tertiary structure. This gives the three-dimensional views of the protein. Tertiary structure is absolutely necessary for the many biochemical activities of proteins. Some proteins are in assembly of more than one polypeptide or subunits. The manner in which these individuals folded polypeptides or subunits are arranged with respect to each other. Linear strings of the spheres arranged in one upon each other in the form of the cubes or the plate, etc., is the architecture of the protein, otherwise called the quaternary structure of the protein. Adult human hemoglobin consists of the 4 subunits. 2 of these are identical to each other, hence the 2 subunits are alpha type and the 2 subunits are beta type together constitute of the human hemoglobin Hb. Nature of the bond liking monomers in the polymer. In the polypeptide or the protein, amino acid are linked by the peptide bond, which is formed when the carboxylic group is one of the amino acid reacts with the amino group of the text amino acid with the eliminations of the water moistly. In the polysaccharides, the individual monosaccharides are linked by a glycosidic bond. This bond is also formed by the dehydration. This bond is formed between the two carbon atoms of the two adjacent monosaccharides. In the nucleic acid, a phosphate moiety links in the 3' carbon of one sugar molecules 1 nucleotide to 5 nucleotide carbon of the sugar of the substituting nucleotide. The bond between the phosphate and the hydroxyl group of the sugar is ester bond, and there is one of the such ester bond on either side. It is called phosphodiester bond. Nucleic acid exhibit the wide variety of the secondary structure. For the example, one of the secondary structure exhibited by the DNA is the famous Watson and Crick model. This model says that DNA exists as a double helix. The two strands of the polynucleotides are antiparallel, that is run into the opposite direction. The backbone is formed by the sugar phosphate sugar chain. The nitrogenous bases are projected more or less perpendicular to this backbone but face inside. A and G of one of the strands compulsorily base pairs with T and C respectively. On the other strand, there are 2 hydrogen bonds between the A and T and 3 hydrogen bonds between the C and G. Each strand appears like a helical staircase. Each step of the ascent is represented by a pair of bases. At each step of the ascent, the strands turns 36 degrees. One full turn of the helical strands would involve the 10 steps or 10 base pairs. Attempt drawing a line diagram. A pitch would be the 34 angstrom. The rise per base pair would be the 3.4 angstrom. This is from the DNA with the above mentioned silent features is called the beta bDNA. In higher classes, you will told that there are more than the dozen forms of the DNA named after the English alphabets with the unique structural features. Dynamic state of body constitute concept of the metabolism. What we have learned till now is that the living organism, be it a simple bacterial cell, a protozoan, a plant or an animal, contain the thousands of the organic compounds. These compounds or biomolecules are present in the certain concentration, expressed as a mole, cell or mole per liter. One of the greatest discoveries ever made was the observation that all these biomolecules have a turnover. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and making is through the chemical reactions constantly occurring in the living organisms. Together, all these chemical reactions are called metabolism. Each of the metabolic reactions result in the transformations of the biomolecules. A few examples for such a metabolic transformations are removal of the CO2 from amino acid, making an amino acid into an amine, removal of amino group in the nucleotide base, hydrolysis of the glycosidic bond of the disaccharides, etc. We can list 10,000 of such examples. Majority of these metabolites reactions do not occur in isolation but are always linked to some other reactions. In other words, metabolites are converted into each other in the series of linked reactions called metabolic pathway. These metabolic pathways are similar to the atomic traffic in city. The pathway is either linear or circular. These pathways crisscross each other and there are the traffic junctions. Flow of metabolites through the metabolic pathway has a definite rate and directions like automobiles traffic. This metabolic flow is called the dynamic state of the body constitute. What is the important is that this interlinked metabolic traffic is very smooth and without a single reported mishaps for the healthy conditions. Another features of these metabolic reaction is that every chemical reaction is a catalysis reactions. There is no uncatalytic metabolic conversion in the living system. Even CO2 dissolving in the water is a physical process, is a catalyzed reaction by the living system. The catalysis which has turned the rate of the given metabolic conversion as are also proteins. These proteins with the catalytic power are named enzymes. Metabolic basis for the living. Metabolic pathway can lead to more complex structure from a simple structure. For example, acetic acid becomes the cholesterol and lead to the simple structure from the complex structure. For example, glucose becomes the lactic acid in our skeletal muscles. The former cases are called the biosynthetic pathway of the anabolic pathway. The latter constitute the degradations and hence are called the catabolic pathway. Anabolic pathway, as the expected consume energy assembly of the protein from the amino acid requires energy input. On the other hand, catabolic pathway lead to the release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscles, energy is liberated. And this metabolic pathway from glucose to the lactic acid which occur the 10 metabolic steps called the glycolysis. Living organisms have learned to trap this energy liberated during the degradations of the store. It is in the form of the chemical bonds. As when we needed this bond, energy is utilized by the biosynthetic, osmotic and mechanical works that we perform. The most important form of the energy currency of the living system is bond energy in the chemical is called the adenosine triphosphate or ATP. How do the living organism derive their energy? What strategies have they evolved? How do they store this energy and in what form? How do they convert this energy into work? You will study the understand all these under a sub-disciplines called the bioenergetics later in your higher classes. The living state. At this level, you must understand that the tens and the thousands of the chemical compounds in the living organism, otherwise called the metabolites or biomolecules, are present at the constructions characteristics of each of them. For example, the blood concentration of the glucose in normal healthy individuals is 4.2 molar per liter, 6.1 molar per liter, which that the hormone would be nanograms ml. The most important fact of the biological system is that all living organisms exist in the steady state of the characteristics by the concentrations of each of these biomolecules. These biomolecules are the metabolic flux. Any chemicals and physical process move spontaneously to equilibrium. The steady state is non-equilibrium state. One should be remember for the physics that system of the equilibrium cannot perform the work as the living organism work continuously. They cannot be afford to reach equilibrium. Hence, the living state of the non-equilibrium steady state to be able to perform the work. Living process is the constant effort to the prevent falling into the equilibrium. This is achieved by energy input. Metabolic provides mechanism for the production of the energy. Hence, the living state of the metabolisms are synonymic. Without metabolism, they cannot be the living state. Almost all enzymes are proteins. There are some nucleic acid that behaves like enzymes. These are called ribozymes. One can depict an enzyme by the line diagram. An enzyme, like any protein, has a primary structure that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and tertiary structure. When you look at the tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself and hence many cervices or pockets are made. One such pocket is the active site. An active site of the enzyme is the creative and pocket into the substrate fits. The enzyme through their active sites catalyze reaction at the high rate. Enzyme catalyzes different from inorganic catalysis in many ways. But one major difference needs mention. Inorganic catalysts work as essentially at high temperatures and high pressures, while enzymes get damaged at the high temperatures. However, enzymes isolated from the organism who normally live under the extremely high temperature are stable and retain their catalytic power even at high temperature. Thermal stability is thus an important quality of such enzyme isolated from the thermophilic organism. Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of the changes. A physical change simply refers to as the change in the shape without breaking of bonds. This is the physical process. Another physical process is the change in state of matter. When ice melts into water and when water becomes a vapor, these are also physical process. However, when the bonds are broken and the new bonds are formed during the transformation, this will be called a chemical reactions. For example, in an organic chemical reaction, similarly hydrolysis of a starch into the glucose is the organic chemical reaction. Rate of the physical and chemical processes refers to the amount of product formed per unit time. It can be expressed as rate into delta P upon delta T. Rate can also be called the velocity if the direction is specified. Rate of the physical and chemical processes are influenced by the temperature, among other factors. A general rule of the thumb is that the rate doubles or decreased by half for every 10 degrees Celsius changes in either direction. Catalyzed reaction proceeds at the rates vastly higher than that of the uncatalyzed one. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, CO2 plus H2O, H2CO3. In the absence of any enzyme, this reaction is slow, with about 200 molecules of the H2CO3 being formed in an hour. However, by using the enzyme present with the cytoplasm called carbonic anhydrase, the reaction speed dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of the enzyme is incredible indeed. There are thousands of the type of the enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction when each of the steps is catalyzed by the same enzyme complex or different enzymes is called the metabolic pathway. For example, glucose convert into 2-pyruvic acid. It is actually the metabolic pathway in which the glucose becomes pyruvic acid through a 10 different enzyme catalyzed metabolic reaction. When you studied the respiration in chapter 14, you will study this reaction. At this stage, you should know that every metabolic pathway with one or two additional reactions give rise to the variety of the metabolic end products. In our skeletal muscles, under the anaerobic condition, lactic acid is formed. Under the normal aerobic conditions, pyruvic acid is formed. In yeast, during the fermentation, the same pathway leads to the production of the ethanol. Hence, in different conditions, different products are possible. How do enzymes bring about such a high rate of chemical conservations? To understand this, we should study the enzyme a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to the reaction. The chemical which is converted into the product is called the substrate. Hence, enzyme, that is the protein with the three-dimensional structure including an active site, convert the substrate into the product. Symbolically, this can be depicted as SP. It is now understood that the substrate S has to bind with the enzyme at its active site with the given cleft and pocket, and the substrate has to diffuse towards the active site. There is an obligate formation of the ES complex. The E stands for enzyme. This complex formation is the transient phenomenon. During the state of where the substrate is bounded to the enzyme active site, a new substructure of the substrate called the transition state structure is formed. Very soon after the expected bond making is completed, the product is released from the active site. In other words, the structure there could be many more altered structural state between the stable substrate and the product. Implied in this the statement in the fact of all other Intermediate structural states are unstable. Stability is something related to the energy status of the molecular or the structure. Hence, when we look at the pictorially through a graph, it looks like something as in figure. The y-axis represents the potential energy content and the x-axis represents the progression of the structural transformation or state through the transition state. You would notice two things: the energy level difference between the S and P. If P is at lower level S, the reaction is an exothermic reaction. One need to not supply energy in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic and energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of the S form, that is this transition state, is called the activation energy. Enzymes eventually bring down this energy barrier, making the transitions of S to P more easy. Nature of the enzyme action. Each enzyme E has a substrate S binding site in its molecule so that a highly reactive enzyme substrate complex is produced. This complex is short-lived and dissociated into its product P, and the unchanged enzyme with the intermediate formations of the enzyme product complex. The formation of the ES complex is essential for catalysis. The catalytic cycle of the enzyme's action can be described in the following steps. First, the substrate binds to the active site of the enzymes fitting into the active site. The binding of the substrate induced the enzyme to alter this shape, fitting more tightly around the substrate. The active site of the enzymes now is close. Proximity of the substrate breaks off a chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme released the product of the reaction and the free enzymes is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Factors affecting on enzyme activity. The activity of an enzyme can be affected by the change in the condition which can alter the tertiary structure of the protein. These include the temperature, pH, change in substrate concentration, or binding of a specific chemicals that regulate its activity. Temperature and pH. Enzyme generally function is a narrow range of the temperature and the pH. Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Actively decline both below and above the optimum value. Low temperature preserves the enzymes in the temporary inactive state, whereas high temperature destroys the enzymatic activity because proteins are denatured by heat. Concentration of a substrate. With the increasing the substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, which is not exceeded by any further rise of the concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after the saturation of these molecules, there are no free enzymes molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to presence of a specific chemicals that bind to the enzyme. When the binding of the chemical stuffs shuts off the enzyme activity, the process is called inhibition and the chemical is called the inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzymes, it is known as the competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding side of the enzyme. Consequently, the substrate cannot be bound with the. As a result, the enzyme action decline and inhibitions of the succinyl dehydrogenase by malonate, which closely resemble with the substrate. Succinate is the structure. Such a competitive inhibitors are often used in the control of bacterial pathogen. Classification and nomenclature of enzyme. Thousands of the enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of the reaction they catalyze. Enzymes are divided into 6 classes, each with the 4 to 13 subclasses and named according to the by 4-digit number. Oxidoreductase dehydrogenase enzyme which catalyze the oxidoreductase between the two substrate S and S dash. Transferase enzyme catalyzing a transfer of the group G between the pair of the substrate S and S dash. Hydrolysase enzymes catalyzing the hydrolysis of ester ether peptide glycosidic CCC halidine PN bonds. Lysase enzymes that are catalyzed removal of the groups from the substrate by the mechanism other than the hydrolysis leaving double bonds. Isomerase include all the enzyme catalyzing interconversion of the optical, geometrical and potential isomers. Ligase enzymes that catalyzing the linking together of the two compounds, that is enzyme which catalyze the joining of CO, CS, CN and PO etc. bonds. Cofactors enzymes are composed of one or several polypeptide chain. However, there are a number of the cases in which non-protein constituent called the cofactors are bound to the enzyme to make the enzyme catalyzing active. In this instant, the protein portion of the enzyme is called the apoenzyme. Three kinds of the cofactors may be identified: prosthetic group, coenzyme and metal ions. Prosthetic group are the organic compounds and are distinguished from other cofactor in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalyzase, which catalyze the breakdown of the hydrogen peroxidase to water and oxygen. Heme is the prosthetic group and it is the part of the active site of the enzyme. Coenzyme are also organic compounds, but their association with the apoenzyme is now only transient, usually occurring during the course of the catalysis. Furthermore, coenzymes serve as a cofactor in the number of the different enzyme catalyzed reactions. The essential chemical compounds of many coenzymes are vitamins. Example: coenzymes nicotinamide adenine dinucleotide and NADP contain the vitamin nitasin. The number of the enzymes require metal ions for their activity, which form the coordination bond with the side chains at the active site, and they same time from the one or more coordination bonds with the substrate. Example: zinc is the cofactor of the prosthetic enzyme of carboxypeptidase. Catalytic activity is most when the cofactor is removed from the enzyme, which testified, and they play the crucial role in the catalytic activity of the enzymes.